How can a computer look at raw DNA and discover genes? In this video, we'll follow the complete journey of the data. Starting from DNA letters, we'll convert sequences into tensors, learn biological patterns with a convolutional neural network, and finally apply Viterbi decoding to produce biologically consistent gene predictions. Let's watch how the data changes at every step. Everything starts with a DNA sequence. At this stage, the computer only sees a long string of letters. Our example contains the 4 DNA bases, A, C, G, and T. Neural networks cannot directly process letters. So each DNA base is converted into a numerical ID. A becomes 0, C becomes 1, G becomes 2, and T becomes 3. One DNA sequence is not enough for training. We combine many sequences into a large tensor. This allows the neural network to learn from thousands of examples at the same time. Each DNA position also has a correct biological label. These labels are converted into numbers just like the DNA sequence. The neural network will learn to predict these labels. Numbers alone are not very informative. The embedding layer converts each DNA base into a learned feature vector. Now, every position contains richer information that the neural network can use. The tensor is now rearranged into the format expected by the convolutional neural network. No biological information changes. We are simply organizing the data for efficient processing. The convolutional neural network scans along the DNA sequence. It searches for important biological patterns and motifs. Examples include start codons, stop codons, and other sequence signals. After the CNN finishes its analysis, the tensor is rearranged again. Now each DNA position contains dozens of learned biological features. These features will help classify each position. The classifier examines the features at every DNA position. It produces scores for all possible gene states. These scores represent how likely each state is. The model compares its predictions with the correct labels. The difference becomes a loss value. The goal of training is to make this loss as small as possible. The loss tells the network what it got wrong. Backpropagation adjusts millions of tiny parameters. With each training step, the model becomes a little more accurate. The classifier scores are converted into probabilities. Now we can see how confident the model is about each prediction. Higher probabilities indicate stronger confidence. The neural network now predicts a state for every DNA position. These predictions come directly from the learned patterns. However, some predictions may still be biologically inconsistent. The Hidden Markov Model adds biological rules. Viterbi Decoding searches for the most likely sequence of states. This helps remove impossible transitions and creates more realistic gene structures. After combining neural predictions with constraints, we obtain the final gene labels. The system can now identify intergenic regions, start codons, exons, and stop codons. I developed a hybrid gene prediction system in Python that combines deep learning and probabilistic modeling. The pipeline converts DNA sequences into numerical tensors, uses a CNN and BLSTM neural network to learn sequence patterns, and then applies Hidden Markov Model Viterbi Decoding to enforce biologically valid gene structures. The project includes synthetic genome generation, model training, validation, and gene annotation prediction. If you're interested, the code is available below the link.